Do you still remember how to answer the experimental questions? We use chromes here. So let's see the question 10 here. Plant absorb light energy for photosynthesis. Some colors of light result in a higher rate of photosynthesis than other colors of light. Devise an investigation to discover which color of light result in a higher rate of photosynthesis. So in this question, we need to identify what is the independent variable and dependent variable. So we say independent variable is the cause and the dependent variable is the result. Here we can see the light color is the cause and the photosynthesis rate is the result, because when we change the color of light, there are different photosynthesis rates, right? So we can say the color of light is the independent variable and the photosynthesis rate is the dependent variable, because we change the color of light in each setup before the experiment, right? So this is the cause and this is the result. So we can identify independent variable and dependent variable based on the cause and the result, okay? So now let's describe how we do the experiment. We say there are 5 directions we need to mention in this type of investigation questions. First of all, C stands for change. So what should be changed before experiment in each setup is the color of light. So we use the light bulbs with different color. That is the independent variable of this experiment. So C. The next one is O. O stands for organism. So which organism is involved in this experiment? For photosynthesis, of course, it's plants. So we try to describe plants. Like we can mention they are the same species in different setup, the same age, the same size, the same condition. So we need to do some description, okay? The third one is R, repeat. But we try to be more specific, not just mention, repeat and then calculate the average. We try to be more specific to this question. Like we can repeat 100 times in each color of light and then we calculate the mean, okay? So what mean we should be measured? So this point is measure. How we measure the rate of photosynthesis, we can use the volume of oxygen produced or count bubbles. So just like what we have mentioned, for example, we do 100 plants under red light and then they have different volume of oxygen produced. Then we measure the volume of each setup and then divided by 100. So this is how we repeat and calculate the mean. Another point for measure is the time we use. For example, we say, let the setup stay there for 30 minutes. Just a reasonable time, but not just 1 minute. That is unreasonable, right? For the endpoint, we can say in 3 directions. One is how we measure. Like here, we can measure the volume of oxygen. Or second, what the tool you used. For example, here, we can use the inverted measuring cylinder because when the gas evolves, you cannot see. But underwater, due to the displacement of water, you can see how much oxygen produced. The third direction, you can talk about the duration of the experiment. So if you describe an experiment, better to say from how you measure, what the tool you use, and the duration of the experiment, okay? Last one, S. S stands for same. It's the control variable. What you need to control in this experiment, we try to mention in 2 aspects. For example, here is the conditions. For example, you need to control the environment, temperature, the light intensity. Make sure the color of light is the only thing changed in different setup. And the other side of the control variable, maybe we can talk about the parameters. For example, carbon dioxide concentration, the pH, the mineral concentration in the soil, that also affect the photosynthesis rates. But one thing you need to be reminded is you need to write in full sentences. So you cannot just follow me in the point form. So try to make it in the full sentences and include all the points here, okay?